Dear students, welcome to Bluepin Online. Shall we begin? Please use headset, pen and paper to take notes.
Here you go. Please let's know your feedback after class.
Specialized membrane structures and transport mechanisms. Okay, we discussed our biological membrane. Now we are entering into specialized membrane structure. I think slide is clear. Yes, you can see the slides, Sandhya?
Yes, ma'am.
Okay. Then the first specialized structure, membrane structure, is tight junction. Okay, tight junction that permits calcium and small molecules to pass through from one cell to another through narrow hydrophilic pores. So the communication between the cell that occurs through this junction. Okay, then absence of tight junction is implicated in the loss of contact inhibition in cancer cells. Only at that time, absence of tight junction occurs. Tight junction that seal off subepithelial spaces of organs from lumen that contain some specialized proteins, occludin, claudin, and adhesion molecule. Okay, adhesion molecule. Okay, so what is tight junction? It's a specialized structure. Now we discuss the specialized membrane structure. Which are the specialized structures present in plasma membrane? Among those, first one is tight junction. Tight junction, that is mainly for the passage of calcium and small molecules from one cell to another through some hydrophilic pores. Okay, and the communication between the cells occurs also with this junction. Only in case of cancer cells, this absence of tight junction occurs that seals off subepithelial spaces of organs from lumen. Some specialized proteins are present in tight junction. Those are occludin, claudin, and adhesion molecule. You can see from this picture, you can see there is a space. This particular area is known as tight junction. You can see the proteins like claudin, occludin. Okay, that forms the tight junctions. Okay, you just note that protein name, occludin and claudina and occludina. Okay, these are the main proteins present in the tight junction. Then what is the role of tight junction? Okay, now we complete the role of tight junction that present as a specialized membrane structure. Okay, next the second one, that is known as gap junction. Gap junction and tight junction both are different. Okay, the second part is gap junction. Most eukaryotic cells are in contact with neighboring cells, and these interactions are the basis of formation of organs. Cells are in metabolic contact. These interactions are called gap junction. Okay, so gap junctions make some interaction between neighboring cells, and this is the basis for the formation of organ. And cells are in metabolic contact. We need contact. Okay, mainly metabolic contact, and these interactions are called gap junctions. Gap junctions ensure continuous supply of nutrients to cell of an organ which are not in direct contact with the blood supply. That is the most important point. Gap junctions ensure continuous supply of nutrients to cells of an organ which are not in direct contact with the blood supply. Okay, then gap junctions are formed by a type of protein. It is called as connexins. Tight junctions, we can see claudins and occludins. Here, gap junctions are formed by a type of protein called connexin. Okay, you can see from this diagram how it is open and closed, this gap junctions. Okay, you can see this gap junctions. Okay, the protein name is connexon or connexins are present. Okay, then here explain the structure of connexin. It's a protein, polypeptide, starting from N-terminal to C-terminal. Okay. Then next, third one, it is very familiar, myelin sheath. Myelin sheath, it is made up of membrane Schwann cells. Myelin that is composed of sphingomyelin, sphingophospholipid, sphingomyelin, cholesterol, and cerebroside. Cerebroside coming under glycolipids. Okay, globoside, cerebroside. Okay, so myelin sheath thin out in certain regions. Now it is nervous system. Okay, myelin sheath thin out in certain region. That particular region is known as nodes of Ranvier. Due to this arrangement, propagation of nerve impulse is wave-like, and the speed of propagation is also increased. In case of disease, multiple sclerosis, demyelination occurs. Then what happens? Velocity of nerve impulse reduced, that leading to motor and sensory deficit. Okay, that's about our myelin sheath. Okay, then microvilli. Microvilli of intestinal epithelial cells and pseudopodia of macrophages are produced by membrane evagination. This is due to the fluid nature of membrane. We are very familiar with that topic, microvilli. Then membranes of organelles. Okay, instead of plasma membrane, we can see membranes of endoplasmic reticulum, nucleus, lysosome, and mitochondria. Okay, percentage of protein content that will be vary in different organelles. Percentage of protein content varies from 20% in myelin sheath to 70% in mitochondria. That is the major difference between plasma membrane and membranes of endoplasmic reticulum, nucleus, lysosome, and mitochondria. Okay, now you can see the structure of villi. What is the role of villi? That increases surface area. Okay, and then all other things we need only the structure like villi. These are the specialized structure. Okay. Now it is the time for interaction. I think the topic is clear. Sandhya? Hello, Sandhya?
Yes, ma'am.
Okay, I think it is clear.
Yes, ma'am.
Okay, what is tight junction?
Junction that permits calcium in small molecules to pass from one cell to another through hydrophilic pores.
Okay, then there are two proteins that is present in tight junctions. Which are they? Name is difficult here.
Claudins.
Very good. Claudins and occludin. Okay. Okay. Then, Andrea, what is gap junction?
They form they have metabolic communication.
Metabolic communication.
Yeah, they form the basis for forming organs. Then the protein present is connexin.
Connexin, most important point in gap junction.
They ensure continuous supply of nutrients to the cells where they do not have enough blood supply.
Okay. Okay, very good, Andrea. Next, Roshni, what is the role of myelin sheath?
Ma'am, the myelin sheath is basically made up of membrane Schwann cells, and it's thin in regions of the node of Ranvier.
Okay.
And yes, ma'am, it provides a sufficient velocity for transport of impulses.
Okay.
And in case of demyelinating, in multiple sclerosis, there's demyelination of the myelin sheath.
Okay, very good. Then Meghna, what is the role of microvilli?
Microvilli, it is for to increase the surface area, and it is formed by the invagination of the membrane due to the fluid nature.
Okay, that is the main point. Fluid nature increases the fluid nature. Then so there are some other organelles that contain membrane. Which are they?
Endoplasmic reticulum, mitochondria,
and
lysosomes.
Okay. Sandhya, what is the difference between plasma membrane and other membrane organelles contain membrane? What will be the major difference, Sandhya?
Membrane in endoplasmic reticulum.
Yes.
The percentage of protein content varies from 20%.
Yes. that percentage of protein content may vary. We know from the chemical composition of plasma membrane, the major content, almost 55% is protein. That protein content may vary from 20% to 70% in different membrane. Okay, that is the difference. Okay, Sandhya, thank you. Then next we are entering into another area. Actually, it's also a specialized structure that is known as cytoskeleton. Okay, structure of cell is maintained by cytoskeleton that is present underneath the plasma membrane. What is the role of cytoskeleton? Cytoskeleton is responsible for shape of the cell, cell motility, and chromosomal movements during cell division. These are an important question is cytoskeleton, short note. So, cytoskeleton is responsible for shape of the cell, cell motility, and chromosomal movements during cell division. Then what are the contents? What are the different areas, different structures present in cytoskeleton? It is composed of microfilaments, intermediate filaments, and microtubules. Okay, there are three structures coming under cytoskeleton, that is microfilaments, intermediate filaments, and microtubules. Then what is this microfilament? That is made up of G-actin found in almost all cells fused to form F-actins that exist as a tangled meshwork of 8 to 9 nanometer in size. Then the second one is known as intermediate filaments. Intermediate filaments, that is 10 nanometer in diameter, rod-like elongated structures. These are the stable components of cytoskeleton. Okay, so we discussed two particles, microfilaments and intermediate filaments. Okay, microfilaments, that is made up of G-actin that is found in almost all cells that fuse to form F-actins that exist as a tangled meshwork. Size is 8 to 9 nanometer. The second part, that is intermediate filaments, that is present in 10 nanometer diameter, that is rod-like elongated structure. Stable components of cytoskeleton is intermediate filaments. For example, keratin. That is a special type of protein, and keratin in hair and nail, and lamins provide support for nuclear membrane. Lamin is another type of protein that provide support for nuclear membrane. These are also coming under intermediate filaments. Okay. Then you can see the structure, low G-actin concentration and high G-actin concentration. Okay. These are the structure of microfilaments and intermediate filaments, rod-like intermediate filaments you can see from the diagram. Okay. Then the third section of cytoskeleton, that is our microtubules. Microtubules that contain tubules, that contain a special protein tubulin. So that contain alpha and beta tubulin with diameter 25 nanometer. Okay. That is essential in the formation of mitotic spindle. That word is very familiar at the time of cell division, mitotic spindle. Okay, so this microtubules, tubulin is involved in the formation of mitotic spindle and participate in exocytosis and endocytosis. Okay, tubulins polymerize to form cylindrical filaments that assemble into sheaths and fibers. Microtubule associated proteins that stabilize the assembly. For example, the abnormal aggregation occurs. Abnormal aggregation of tau proteins are found in degenerative diseases. If there is any kind of abnormality in assembly, for example, the tau protein assembly, that will be causes some degenerative diseases. And in case of degenerative diseases, we can see the abnormal aggregation of tau proteins. Okay, that's about the structure of microtubules, the alpha and beta tubulins. Okay. I think it is clear for you. The cytoskeleton, we discuss microfilaments, intermediate filaments and microtubules. Okay. Then one more thing is there in specialized membrane structure, that is our molecular motors. What is it? Proteins that are responsible for coordinated movements in tissues and cells are referred to as molecular motors. Motors, so they need energy. They are driven by ATP. For example, actin and myosin in muscle and kinesins. Okay, is it speed? Am I going fast? Is it okay for you?
Okay, ma'am.
Okay. Then Santya, then it is the interaction time. Okay. What is molecular motors, Sandhya?
Coordinated movement in tissues and cells.
Okay. Then energy is needed for the action of molecular motors?
Yes, ma'am. ATP.
Okay. What example? Example for molecular motors?
Actin, myosin, kinesin.
Okay, very good. Thank you. Then Andrea, what are the roles of microtubules?
Microtubules, they are made up of alpha and beta tubulin, and their role is they are involved in mitotic spindle formation.
Okay.
They
speciality.
During degenerative diseases, there will be abnormal aggregation of tau protein.
Okay, then there is an assembly. In case of this microtubules, this tubulin, the major protein is tubulin. Okay, the tubulin that polymerize to form cylindrical filaments. Okay, that assemble into sheaths and fibers. Okay, the microtubule associated proteins that stabilize the assembly. Okay, an assembly is essential for this. You can see from the diagram. An assembly is essential for the formation of microtubules, okay, by the condensation of this tubulins, alpha and beta tubulins. An assembly is needed. Some proteins help for this assembly. If there is any kind of disorder or abnormality occurs in that assembly, that will lead to degenerative diseases. For example, in the case of top protein assembly, if there is any kind of abnormality, that will lead to degenerative disorders. Okay, that is the importance of microtubules. Thank you, Andrea. Then next question, that is what about microfilaments, Roshni? Microfilaments.
Ma'am, microfilaments, their diameter primarily ranges from 8 to 9 nanometers. They are composed of G-actin, which fuse to form F-actin, ma'am.
Okay, you can see from the diagram where there is low G-actin concentration and high G-actin concentration. G-actin fuse to form F-actin. Okay, then next is Meghna. What is the role of intermediate filaments? Role of intermediate filaments?
It forms a stable component in the cytoskeleton. It's a rod-like structure.
Yes, from the diagram it is clear. Its intermediate filament is a rod-like structure. Okay, rod-like. Example for intermediate filaments. Example for intermediate filament.
Keratin in hair.
Hair, only hair.
Hair and nail.
Okay, then one more protein.
And lamins.
Lamins, that is present.
That provides support for nuclear membrane.
Okay, very good. Which type of protein is keratin? In protein classification, which type of protein is keratin? Anyone?
Structural
protein. Okay, very good. Okay, then next we are entering into interaction. Now the interaction time. If you have any doubt, you can ask me now. Any doubt? Is it clear with all our topic? Are you okay? Can you follow me? Yes or no?
Yes, ma'am, it's clear.
Okay. No doubt. If you have any doubt, we are entering into the other topic. So if you have any doubt about the specialized structure of membrane, you can ask me now. We complete tight junction, gap junction, then cytoskeleton and all other things. So now we are entering into transport mechanism. Biological membrane is over now. Next topic is transport mechanism. Okay, I think there is no doubt. Is it clear? Roshni?
Yes, ma'am, clear, ma'am.
Okay, Sandhya?
Yes, ma'am, clear.
Okay. Then next is, we are very familiar with the transport mechanism from +2 classes onwards. We are very familiar, but now in biochemistry, clear-cut knowledge is essential for the transport mechanism, because all our metabolic pathways and all our things are based on this transport mechanism. Okay, so the permeability of substances across the membrane that depend on the solubility in lipids and not on their molecular size. Okay? Water soluble molecules are impermeable and require carrier mediated transport. Then, membrane always withhold unwanted molecules and permitting entry of molecule necessary for metabolism. So, this transport mechanism play a very important role in our body. In biological membranes, all biological membranes have this transport mechanism. That is one of the main function, one of the major area carried out by this transport mechanism, because the membranes, the permeability of substance across the membrane is dependent only on the solubility in lipid, not the solubility in water. Water soluble molecules are impermeable because lipid bilayer membrane. So it is completely dependent on the solubility in lipid, not on their molecular size. If it is small, very small, or very high, huge compound, no matter, that is not depend on the molecular mass and water solubility. It is strictly dependent on lipid solubility. And sometimes they need a carrier mediated transport. Then one major thing, only unwanted things are withhold. Membrane always withhold unwanted molecules. Okay, in our character, the same mechanism must be there. Okay, membrane always withhold unwanted molecule and permitting the entry of molecule necessary for metabolism. Okay, only the materials which are essential for metabolism that will enter into plasma membrane. All other compounds, all other molecules are withhold. Okay, that's about the introduction about transport mechanism. Next, we classify transport mechanism. Simply we classify. We are very familiar with active transport and passive transport. Which are the other areas coming under passive transport? Simple diffusion, facilitated diffusion, and third one, ion channels. Then in case of active transport, what are the areas that we will discuss later? Then third one is pumps. Okay, so that's about the classification of transport mechanism. Passive transport, active transport. Passive transport, simple diffusion, facilitated diffusion, ion channels. Active transport, and third one is pumps. Okay, now we are entering into passive transport. Okay, what are the importance of passive transport? Passive transport, what is meant by passive transport? There are two areas, simple diffusion and facilitated diffusion. What is simple diffusion? Okay, very simple process. Solutes and gases enter into the cell passively. That is driven by, here it is driven by the concentration gradient. Rate of entry, that is proportional to the solubility of the solute in the hydrophobic core of the membrane. Simple diffusion, that occurs from higher to lower concentration. No need of energy, does not require energy, but it is a very slow process. Okay, these are the 6 points coming under simple diffusion. Solutes and gases that enter into the cell passively. It is driven by concentration gradient. Rate of entry is proportional to the solubility of that solute in the hydrophobic core of the membrane. Simple diffusion occurs from higher to lower concentration, that does not require energy, and it is a very slow process. You can see from the diagram. Okay, there is here cytoplasm, here is the extracellular fluid. What happens? From the higher concentration to lower concentration, the entry occurs. It is known as the simple diffusion method. Then, next, diffusion of gases such as oxygen, carbon dioxide, NO, and CO dependent upon concentration gradient. Lipophilic molecules also diffuses across the membrane at a rate that is directly proportional to the solubility of the compound in the membrane. Okay, I think you are clear with simple diffusion. Okay, is it okay, Sandhya? Hello?
Yes, ma'am.
Okay, no doubt about simple diffusion. It's a very simple topic. Okay, then next is about facilitated diffusion. What is facilitated diffusion? Before that, I want to ask questions. Otherwise, we can't follow this simple. After simple diffusion, we need some questions. Okay, okay, Sandhya. What are the importance of this transport mechanism? Why we need a transport mechanism? Sandhya?
The permeability of substance across the membrane dependent on solubility of lipids. And water-soluble molecules are impermeable.
Okay.
So they require some mediated protein.
Carrier mediated transport. Okay. Then what is the role of molecular size? Is there any role for molecular size? Yes or no?
No.
It is dependent on molecular size? No, it is not dependent on molecular size. Then what is the major function of plasma membrane? Unwanted particles. What happens to unwanted particles that will enter into membrane or withhold? Sandhya? Hello?
Yes, ma'am.
Okay. What happens? The last point of the transport mechanism. What happens to the membrane? Always withhold unwanted molecules and permitting the entry of molecules necessary for metabolism. Okay, Sandhya? Okay. Yes. What are the classification of transport mechanism? Can you name it? What are the classifications?
A simple diffusion, facilitated diffusion.
That is coming under which category?
Passive.
Okay. Then what about the second one is active transport and the last one is pumps. Okay. Then Roshni, what are the points coming under passive transport?
Ma'am, passive transport, basically the solutes move across the membrane from higher concentration to lower concentration. They do not require energy, and solutes and gases pass by passively. And the rate of diffusion is directly proportional to their solubility in the hydrophobic pore.
Okay.
The membrane, and they don't require energy.
It's a very slow process.
Yeah.
Okay. It is not too fast. It's a slow process. Okay. Okay. That's about our simple diffusion. Okay. Then diffusion of gases like oxygen, carbon dioxide, NO, and CO. That depends upon the concentration gradient. Lipophilic molecules also diffuses across the membrane at a rate that is directly proportional to the solubility of the compound in the membrane. Then next we are entering into facilitated diffusion. Okay. This is a carrier-mediated process. Important features of facilitated diffusions are, there are 4-5 points. The important features. The carrier mechanism could be saturated, which is similar to Vmax of enzymes. Structurally similar solutes can be competitively inhibit the entry of solutes. We know what is competitive inhibition. Okay. So the structurally similar solutes can be competitively inhibit the entry of solute. So facilitated diffusion can operate bidirectionally. Okay. It is not a unidirectional process. It can be bidirectional. That does not require energy, but the rate of transport is more rapid than simple diffusion. Simple diffusion is the most slow process. The rate of transport is more rapid than simple diffusion. Okay. Once again, this is a carrier-mediated process. Then carrier mechanisms could be saturated, which is similar to Vmax of the enzymes. Structurally similar solutes that can be competitively inhibit the entry of solute. Facilitated diffusions can operate bidirectionally. That does not require energy, but the rate of transport is more rapid.